Hello, everyone. Today I wanted to continue our discussion on experimental design and get you ready to really be writing that first draft of your research proposal. So again, always to orient us to what our goals are this semester, we're spending time getting ready to develop these research proposals. And right now we're really focused on research methods and coming up with ways to test the hypothesis that we're interested in. So at this point, I hope that you have determined your hypothesis, or at least some version of a hypothesis that is related to the research question you're excited about. I hope that you are working on identifying some of those independent and dependent variables that are related to your hypothesis. And now that you've done that, you're ready to decide on the methodology, how you're actually going to collect the data that will allow you to determine if your hypothesis is supported or rejected. And we talked last time about the difference between experimental studies where you're manipulating some aspect of the study, or purely observational studies where you're just observing organisms in their environment, either natural or captive, but you're not manipulating anything, you're just observing their natural behaviors. So depending on your hypothesis, you may choose either experimental or observational or some combination of the two. To remind you, I'm also busy working on my research proposal, and I came up with the hypothesis that my null hypothesis is that there is no relationship between the sex of the individual, the social situation that they are in, and their stress levels. And I've been working on refining and defining my variables. So obviously when I'm talking about sex as one of my independent variables, I'm talking about males versus females. We know that there are many sex differences in behavior, and so I'm interested in really focusing on if there are sex differences in these behaviors or these responses. I'm interested in social behavior, so I'm going to be looking at fish that are alone or fish that are part of a group. I'm still working on defining exactly what that means, and that's okay. We're always working on defining these variables. And then for my dependent variables, the data I'm going to collect, I'm interested in taking cortisol levels because cortisol is closely correlated to stress levels. And so I'm working on protocol to figure out how to do that. And behaviorally, as I mentioned in the last class, I think I'm going to be looking at swimming activity, since we know that that has something to do with stress in the specific animal, the zebrafish that I'm looking at. So hopefully you all are sort of thinking through that too, thinking what your hypothesis is and what your variables are. You don't have to have it set in stone right now, but just start thinking about what it is that you are going to be manipulating or controlling in your independent variables, or what groups you want to be viewing. And then start thinking about the data that you're going to collect. And I mentioned last time that we'd really get into how to set this up now that we define our variables, because the variables give us a clue as to sort of like the measurements that we're going to have to determine. And so today I want to briefly just talk about ethograms, which we mentioned last time, go through different types of measures of behaviors that you can take, of course, looking at the difference between behaviors that are more states versus events, and then looking at the sampling rules and recording rules and evaluating the pros and cons of each, so you can start thinking about these in terms of what would work for testing your variables as related to your hypothesis with the organism that you're interested in. So everyone will probably look at these differently based on the specific research design that they need for their specific hypothesis. What I want you to do as you're going through this recording today is to take some notes on these with the intent that we can talk about these during our next class. So I'd like you to start thinking about what types of measures you could take if the behaviors recording are states or events, how you might sample your samples, how you're going to record your data. So start thinking about those things, and Tuesday we'll have a discussion about those. So take notes as we're going through this right now, and bring those notes with you so that we can talk about these as related to your projects. So in describing behaviors and coming up with those ethograms, those tables or descriptions of the behaviors that we are going to observe during our study, whether it's observational or experimental, we want to think about certain aspects of the behavior. The first aspect is the structure, right? This is just the appearance, the physical form, the timing of it. For example, if we were looking at dogs, we could look at what the dog was doing, and we could just describe the structure of this behavior. Say he's standing on all fours, he's flexing his front legs so that they're almost horizontal and rest partly on the ground, while the back legs are extended. So you can almost picture this behavior based on this description. We also want to include in our description of behavior the spatial relations, how our organism that is performing this behavior is in relation to other animals or to other structures. So are they approaching something? Are they leaving something? Are they making physical contact with someone or something? Are they breaking physical contact with someone or something? So sort of the location, spatially, where is that behavior occurring? And then we can add in the consequence, so the effects of the subject's behavior on another organism, another animal, the environment, or self. So for example, we can sort of describe the behavior as what it is, right? Like preening versus running the tip of the bill along the primary feather of the wing. So if you know what preening is in birds, it literally is running the tip of the bill along the primary feather of the wing. Excuse me, preening is the term that we use for it, and preening has sort of an intention associated with it because we know that it's used for cleaning and used in social interaction. So it's easier to say preening and knowing what that is, but you do have to define what the behavior itself is. You don't want to wrongly assume what the behavior is for. So you don't want to assume that a vocalization is necessarily a distress call, because it may not be. However, in an organism that has very well-defined behaviors, distress call may be enough information about the behavior to actually describe it and give some sort of intent to it. So when you are describing your behaviors, make sure that you are actually including as much information as you can about those behaviors. Don't just say aggressive behavior. Don't just say play behavior. Describe the physical movement, structure, anatomy of the animal, and the spatial relationship and some consequence of it. So give as much information as you can about that behavior without making any assumptions. Again, I know that you've talked about ethograms in other courses, but to remind you, this is really just a list or a table that contains the precise definitions of species typical behaviors. Ethograms may already exist for the animal that you're interested in, specifically related to the behaviors that you want to study. And so you can look for these throughout the literature, or you may want to come up with your own definitions for the behaviors that you're watching. What's important is that each behavior is operationally defined. You are giving concrete definitions of what a behavior is. So you can call a behavior an approach, but what does an approach really mean? And you'll define that operationally as movement that results in being less than 2 meters from a conspecific for more than 5 seconds. That would be considered an approach. Maybe in opposition to that, you could have a chase. A chase would be movement that results in being less than 2 meters from a conspecific for less than 1 second, because that other conspecific would be moving away. So you can see that both of those behaviors are very similar, approaching versus chasing, but operationally you're defining what each one is. Another example we have here, foraging. We know foraging, you're like, yeah, I know what foraging is. You're eating food. Well, kind of. Foraging is really searching for food, and organisms, different animals, are going to do that differently. So maybe you're looking at some primates. You would define foraging as moving hands through the grass or foliage and ingesting some of the items acquired while moving hands through the grass and foliage. If I was looking at this in my fish, when they forage, they actually flip leaves upside down with their mouths. So the adults will go down to the bottom of a stream, if I'm talking about convict cichlids, and they will flip over leaves and sort of feed on the detritus that's found there. So foraging would be a similar behavior between some primates and some fishes, but it'd be different, and we'd have to operationally define that for each species. This is important for a lot of reasons, and one of them is that a well-written ethogram will allow for reliable observations both within your own observations and between observations of different researchers. So you want strong inter and intra-observer reliability. You want to be able to make sure that every time that you're looking at that animal, you're recording your behavior in exactly the same way. And if there are other researchers, if you have assistants, you're part of a team. I've worked on the same species, the same population of desert pupfish for almost 20 years now, and there are many scientists that work on that population, and we all need to define the behaviors exactly the same. And so it's important that we have a standard ethogram that everyone uses so that we're all recording the same data. Every couple of years we have some issues where data is recorded differently, and so those data need to be treated differently if we're not recording them in the same way. And so it's important to keep that in mind, that an ethogram really defines the behavior that you're interested in and allows you to be consistent, and it also allows others to be consistent. Here are some examples we can think about when we're defining our behaviors. So think about this on your own. Which of these are good ethogram definitions? Let's talk about grooming as a behavior in rhesus macaques. So in macaques, if I wanted to describe the term grooming, would I say it's making physical contact with another individual? Would I say it's running mouth or fingers through the hair of another individual? Or would I say it's running fingers from one or both hands through the hair of another individual for more than 5 seconds? It often involves removing dead skin and parasites from the individual being groomed, and groomer may consume such particles. Which one of those do you think would be the best operational definition for our ethogram? What about if we're looking at wrestling in Belding's ground squirrels? So cute. So Belding's ground squirrels, I want to define what wrestling is. Is it rolling around on the ground? Is it rolling around on the ground with another individual? Or is it facing another individual, often in a ventrum-to-ventrum clench or embrace, and biting at the juvenile's neck, throat, cheeks, or abdomen without inflicting bite wounds? Which one of those do you think is going to be the best operational definition? Keep those in mind when you are defining your behaviors. Another thing you need to consider when you're preparing to start your study is being able to identify individuals. So as we're recording, and we'll talk about the different ways we're going to record and sample our behaviors, sometimes we want to be able to tell the difference between individuals. And so again, this is going to be very species specific, and you're going to have to determine how it will work for your species. Here is just a list of ways in which we often will look at a population like these macaques and try and tell individuals apart. You can use like rings, collars, tags, bands, things that have been attached to the animals, usually things that are different colors. You can tattoo or paint, dye the individuals. Sometimes we use ear notches, or I know in turtles, they'll actually notch the shell of the turtle so that you can tell them apart. So they kind of take a little notch out of them. We'll do that in, we'll also do fin notches when we're looking at fishes. Dyes can be either hair dye if we're talking about mammals, or often we use, in fish and in turtles, we use elastomer tags. So they're almost like little pieces of plastic that go just slightly under the skin, and they're bright colors so you can tell individuals apart. You can also use natural markings. I know when I did work with killer whales, we can actually identify each individual killer whale in a population based on their saddle patch, which is that white swoopy mark that's behind their dorsal fin. Each individual has their own pattern. Humpback whales, the tails all have their own pattern on them, and scientists have been studying populations for so long that they actually can photo identify the different individuals. So that works if you're working with a population that's been well studied for many years. But it can also work in populations that you're just starting to study. I know even with my fish, I work with some coral reef fish called damselfish. Individuals do have slightly different markings. They're highly territorial, so they don't really move very much. But as you look from day to day and see the different males on their territories, you notice that each one has their own individual markings, so you can tell them apart pretty easily. You can also get very fancy, use radio transmitters. These are becoming actually much cheaper and easier to use using GPS and some pretty great technology. But you have to figure that out for your study. If you're going to want to compare individuals, you'll have to figure out how you're going to tell the difference, especially if they're part of a larger social group, and you're not, like me, just picking one fish tank out of the tank at a time. So keep that in mind as you're preparing those ethograms, how you're going to identify the different individuals who are doing the different behaviors, and also who the behavior is being directed at. So you'll want to keep track of those as you're collecting your behaviors. Now, looking at behaviors, we sort of have 4 different types of measures that are associated with the different types of behavior. So we can look at latency of a behavior, the frequency of a behavior, the duration of a behavior, or the intensity of the behavior. These are pretty intuitive, but I'll quickly go through each one and talk about exactly what we mean by each of these terms. But again, they're pretty intuitive if you're thinking about the behaviors that the different animals are doing. So latency is a common way to measure behavior. And latency is really just how much time passes before a behavior occurs. This is measured in units of time. Does it take seconds, minutes, hours, days, right? Like latency to approach. How long did it take that bird, was that zebra finch, to approach the other bird? How long? Did it take 20 seconds? Did it take 95 seconds? Did it take 5 minutes? Did it take 22 minutes? Did it never happen? Obviously, this happened pretty quickly in just a few seconds. But sometimes we'll measure, latency to come out of a structure, latency to inspect something, latency to, I measure latency to retrieve offspring that have been displaced from the nest. How long does that take? Sometimes it takes 5 seconds. Sometimes it takes 5 minutes. It really just depends on the situation and the animals that we are looking at. And so this is sort of one measure because it happens one time, and once you've done it once, that's generally how we measure latency. And it has, some unit of time associated with it. Pretty straightforward. Frequency, also pretty straightforward. How often does a behavior occur? How frequently does it occur? This is usually measured as a rate, as the number of times that the behavior occurs per the length of observation. So for example, how many times per minute does a rat press a lever? How many days per summer is a tiger shark tracked within 5 miles of a particular beach? So how frequent is this behavior? Duration is looking at how long a behavioral pattern lasts. This is usually written as a proportion or a percentage. So again, looking at sort of the total time that you're observing these animals. Out of 30 minutes, monkey A spent 15 minutes grooming other monkeys. You could say that as monkey A spent 50% of her time grooming other monkeys because it was 15 minutes out of 30. You could say that proportionally 0.5, monkey A spent grooming other monkeys was 0.5, the proportion of the time observed. So we want to know of all the time, how much of it, how long did this behavior last? This gives us just sort of a different perspective on a behavior. We can also look at the duration based on how many times the behavior occurred. So sort of looking at duration in terms of frequency of the behavior. So mean duration is really just the total duration divided by the number of times that behavior occurred rather than the total time observed. So again, going back to grooming in monkeys, if 3 grooming bouts occurred and they were 3, 5, and 7 minutes in length, the mean duration for any particular grooming bout was 5 minutes. So that is another way that we can measure duration of a particular behavior. Frequency and duration are not always correlated with each other. And that makes sense. Just because you have a high frequency doesn't necessarily mean you will have a high duration. You might, but you may not. You can have a high frequency with a short duration, where in this example, we're looking at, a parakeet who's engaged in preening another parakeet, and it happens 30 times in 1 hour. So let's say that's a pretty high frequency, but the duration is relatively short. It only occurs for about 5 seconds at a time. 2.5 total minutes out of that whole hour. Conversely, you can also have a low frequency with a longer duration. So that same parakeet on another day may have engaged in preening parakeet B twice in 1 hour, so a low frequency. But those few preening periods lasted for a long time, so 28 minutes at a time. So that's pretty much the entire duration of the observation. So low frequency with a long duration versus high frequency with a short duration. Of course, you could also have high frequency and long durations also, or short frequency, low frequency and short duration. So these aren't always correlated, so it's often important to take both of these measures, as well as intensity. Intensity is our final type of measure where we're looking at the strength or the amplitude of a behavioral pattern. So some examples of this are like looking at foraging, but looking at how much food was eaten. So if you put out 50 grams of a particular food, did they eat all of it or did they eat a little bit of it? That's sort of the intensity of the behavior. How much water did they drink? They jumped, but how high are they jumping? They're walking, but how quickly are they walking? The number of steps that they're taking per minute. If we're looking at some vocalizations, what is the amplitude of that? So intensity gives us another dimension of the behaviors that we're observing. So it's important to include those when possible in those ethograms. Now, looking at those 4 different types of behavioral measurements, we also want to look at the behaviors of interest and determine if they are events or if they are states. And what I mean by that is that some behaviors have short durations. They happen very quickly. And we typically measure those by frequency because they don't happen for a prolonged period of time. They're instantaneous, and so we're counting how many of them there are. For example, the number of tongue flicks per minute, the number of lever taps per minute. States, on the other hand, are behaviors that have longer durations. They're prolonged, and so to measure them as events may not be as informative as measuring them as states. So things like sleeping or foraging or grooming or playing or sitting in proximity to someone else, you might want to have a good measure of how long that is happening. Usually with states, behaviors that have long durations, we measure both the duration and the frequency of that, like we did in the example with the preening parakeets. Looking at the frequency and the duration give us a better idea of how often behaviors are happening and how long they occur. So again, I urge you, look at your behaviors of interest and determine, are these behaviors that I must measure as events, that I just count how many times they happen, or could I learn more information if I measure them as states so that I get an idea of how long a behavior occurs? So these are all things to keep in mind as you're making that list of behaviors. Now, some are going to be really straightforward. You know, how many times? For me, I look at spawning events. How often do they spawn? Those are pretty quick. And we count how many happen. They don't last for more than a second or two. If I want to count the number of bites, a bite happens pretty quickly. But I could measure how long they're engaged in bites and displays. And so that would be more of a state, how long they're doing it. Or how long a male and a female are within close proximity to each other. So within 2 body lengths of each other. That's a state. So like I said, some of those are pretty straightforward, but others are not. Like scratching, sitting, walking, approaching. Are you going to measure those as states or as events? It really depends on your study and the information that you need to gather to examine your hypothesis. So really you need to spend some time thinking about what information would be better to collect about these behaviors that I'm interested in, event data or state data. All of these measures and events and states should be included within your ethogram. So here's an example of an ethogram that we have. You can see that some behaviors are being measured as states. So, how long they're happening for, and others are being measured as events. They've given a code. You don't have to do this, but it's a good shorthand way to record your behaviors. You can give a code for the specific behaviors that you're interested in, and then give your long descriptions and determine if you're going to be looking at those as events or states. Okay, so now you've got it. You've got your ethogram, or you're starting to get your ethogram. You're working on it. You're working on those behaviors that you're going to measure. Now, how are you going to measure them? That's the next question. How do you record behaviors systematically? We need to collect quantifiable behavior if we want to understand the relationships of our variables. And so we have to think about the best way to sample and to record those behaviors that we are interested in. And there's a lot out in the literature about how we can do this. And so we're going to look at 4 sort of different options for sampling and 2 different options for recording our behaviors. So looking first at sampling, we'll look at ad lib, focal, scan, and behavioral sampling. For ad libitum, so ad lib, it basically just means recording behaviors that are visible and are of interest. So watching your population and recording things that happen, writing them down. There are some pros and cons to ad lib sampling. Pros is that you capture really rare but important behaviors because you're just watching and you're just writing down important or interesting behaviors that happen. This is especially helpful during your preliminary observations. So in a real life research proposal, you would be spending time watching your organism as you're developing that ethogram so that you can better define your behaviors. And sort of an ad lib sampling period during that time is important so that you get an idea of the common and the rare behaviors that your specific animal is exhibiting. Now, the cons of this is that it's really biased towards individuals in your population that are the most obvious, the most conspicuous, so the ones that are doing the most behaviors. We're going to record their samples most often. And those behaviors that are most obvious and conspicuous, we're going to be recording those behaviors most often. So this might be useful early on in an experiment, though it may not be how you collect the final data once you have your ethogram and your experiments sort of all set up. Focal animal sampling is very common in terms of behavioral research, where we pick one animal and we watch that one individual for a given period of time. So even if we're looking at a social population, we're looking at a group of macaques, and there's 5 or 10 of them, we would choose just one of those individuals randomly. I don't want you to say, oh, look, we're going to look at the tall one because the tall one's doing something cool. No. You randomly pick one animal as your focal animal for that set time period. Maybe it's 5 minutes or 10 minutes. Could be 3, could be 20, whatever works for your specific animal. But you set a time limit. You pick one animal and you follow that one animal for those 10 minutes. I don't mean physically follow. I mean you watch that one for that time period, and you record all the behaviors of interest. So you look at your ethogram with all those behaviors, and anytime they do one of those behaviors, you record it, or you record the duration of all the behaviors that they're doing, just for that animal. There are pros and cons to this. Pros, one, we get detailed behaviors of individuals. So here, right, I have just like a little sampling of one individual. So this is, S54. We've given her a number, and we've watched her on many different days for 30 minutes at a time, and we've recorded all her behaviors. So the following rows after this, or the following columns after this, would be all of her behaviors that are occurring over these 16 different sample periods that happened over a few months' time. So we're getting detailed behaviors of individuals, which is great. They're also slightly unbiased, since we are just randomly watching that individual at a random time. We're not specifically cueing in on when they're most upset or most angry or most, courty when they're busy reproducing. We're just picking random times to study their behavior. So there's some good reasons why we'd want to do focal animal sampling. There are some cons, though. In the field, it can be difficult to follow one individual. If they're going off on their own, you wouldn't walk after them. You wouldn't want to influence them. So you might not be able to watch them for the entire sample period. The other con, which I did not list here, but the other con is that you may miss interesting behaviors of the other animals that are happening. So if you're only focused on one animal, you're not recording the data of the other animals. It's only the focal animal whose behavior you are recording. And so other interesting behaviors may be happening, which you would have caught in ad lib sampling, but you would not catch in focal animal sampling. So again, pros and cons with this type of sampling method. If you want to catch some of those interesting behaviors, another option for sampling would be scan sampling. Again, this works very well when you're looking at social animals where you have lots of them together. And you basically just census the entire group at set intervals. So you're just counting what they're doing for given periods of time. Generally, you only record a few behaviors that you're interested in. So not everything that everyone is doing, but you're just scanning and saying, okay, how many individuals are foraging right now? How many individuals are sleeping right now? Who is everybody sitting next to right now? And so at some set interval, you're taking these limited measures that are somewhat related to your hypothesis. The pros of this, it's very easy to compare between groups if you're interested in different populations. It's easy to track group movement. Excuse me. The cons of this, some behaviors may be missed. Again, if you're only recording the behaviors of interest, and less conspicuous individuals won't be noticed as well when you're scanning just the group. Those individuals that aren't the most often get overlooked in scan sampling. And then you just have behavior sampling, which I know sounds crazy because all of these are looking at behavior, but behavior sampling is when you're focused on just one behavior, one conspicuous behavior. You're watching the entire group, the entire social group, and you're recording each occurrence of that specific behavior. And you would record the individuals that are involved in that behavior. So who tends to engage in geophagy, which is eating dirt, right? Who is being aggressive at the bird feeder? It's those blue jays, isn't it? It's always those blue jays. I love them, but they're mean to the other birds. So looking though, you're just looking for the number of times that they would chase away another bird. That's the only behavior you're recording. And you're watching everybody and you're just recording those specific behaviors. You're saying the blue jay chased away the black-capped chickadee. The blue jay chased away the house finch. So behavioral sampling is pretty focused. That's good for rare but important behaviors. But the cons are very similar to with scan sampling. You're going to notice these conspicuous behaviors in individuals, and more subtle behaviors will often be missed, and more subtle individuals will often get overlooked. Regardless of what type of sort of sampling that you're doing, you also want to think about how you're recording your data. Is it going to be continuous? Are you just going to write it all down? If you're watching a focal individual, are you just going to watch them for 10 minutes and write down every single behavior that they do? If you're doing scan sampling, are you just going to continuously scan and write down every single behavior that you see? That's continuous recording. There's some really good reasons to continuously record behavior for a given observation window. The one most important thing is that you can record the exact behaviors that are happening, and you can get the exact frequencies and durations for your observation period. If you're watching for 10 minutes and they are foraging for 8 minutes and they are engaged in aggression for 2 minutes and they bit another fish 7 times, you would capture all of that in continuous recording. You'd be able to capture all of that as you recorded all of that data, but there's some cons to this. If you're doing continuous recording, it's usually for a set observation window. But behaviors can happen outside of that window that you're not recording. So again, this is like when you're looking at a focal individual and another individual does something cool. You've missed it. If you're recording and this happens to me so often, I will continuously record the behavior of a male fish, and just as my observation period is ending, a female fish will show up in his territory. And I know they're going to spawn, but I won't get to record that spawn as part of my behavioral data because my observation is done. And so, you have to follow those rules. So sometimes continuous recording can be frustrating. Well, he was probably frustrated too, but it can be frustrating. If the animal disappears, walks away, swims away, that also ruins your continuous recording period, especially if you have a set amount of time that you must watch for according to the rules that you set up. It's also very demanding. It's difficult to write down everything that they're doing. You get better at it with practice, but those first few times that you're observing is very difficult. And you have to really focus on specific behaviors that are part of your ethogram and not necessarily everything that they're doing. Or else you're not able to realistically capture that data. And so your reliability will suffer, right? We talked about inter and intra rater reliability. That will suffer if there's far too many behaviors, because you'll think to yourself, was that a bite? Was that a display? Was that a chase? Was it an approach? What was it? You can't really analyze it because you're continuously recording data. If you stop to think about it, they're already doing their next behavior. So you have to constantly do it. So it's difficult. There are lots of cons, but there are also the pros of getting concrete, discrete measures of the behaviors that you're interested in. Alternatively, you can do instantaneous sampling. So this is sampling that occurs at a given time point or within a specific time range. So there's sort of two ways to do this. The first is called point time sampling, where you're essentially taking a snapshot of the behavior every 15 seconds, 30 seconds, 1 minute. You want it, or maybe even once an hour. You go and you observe this behavior occurring and record it in that way. So, for example, you set a timer for 30 seconds. You sit down, you're looking at all of your macaques that are walking around, and 30 seconds later, a beep happens. And then you look and see where everybody is and you write it down. Right? So kind of like scan sampling, but this is happening at a set time point. So every minute you're taking an observation of that. Every hour, my research colleague would note where a fish was in a tank. So she had fish in tanks and she was trying to figure out if females preferred, this male or that male. And this was over a long time scale because the fish that she was studying have these prolonged courtship periods. And so she would look multiple times a day, once an hour, and say, okay, she's on the left side with this male, or she's on the right side with that male, or she's in the middle. And this would go on for days and days. That would be point time sampling. So it's looking at usually a location or a specific posture or, proximity to someone, because states tend to work better for this than events. If you're looking at events, how often is this behavior happening? It's difficult to capture within point time sampling. That makes sense, right? It's difficult to capture those event behaviors at a specific moment. So an alternative to, before I get to the alternative, I sort of went through these pros and cons already, right? Pros, you can calculate a proportion of time that something's happening. So in my example with the fish, we could say that out of 100 hours of sampling, 75 of those, the female was with a specific male. So we know that she spent the majority of her time with that male. But as I said, there are some cons. You miss the rare behaviors. It's very difficult to capture events rather than states. There's observer bias because you may want to write down something that happened right before the beep or right after the beep, but you can't. It doesn't give us a true picture of what is going on, and it won't necessarily generate a picture of the sequence of the behaviors that are happening like continuous sampling would. But I was going to say an alternative to this point interval capture where you're listening essentially for the beeps and saying what's happening right as this beeps, right at this 15-second mark, is an interval sampling. We call this 1-0 sampling, where if during that 15-second interval a behavior did happen, we would score that as a 1, and if the behavior didn't happen, we would score that as a 0. So we'd watch for 15 seconds, the beep would happen, and we'd say, okay, spawning happened within those 15 seconds. We'll mark it as a 1. We'd be watching for the next 15 seconds. Beep, spawning happened. We'll mark it as a 1. We watch for 15 more seconds. Beep, spawning didn't happen. We mark that as a 0. This is good in some ways because it allows us to capture behaviors that start and stop frequently and tend to be of short duration so that we're not continuously, start, stop with our timer wanting to know what's going on in continuous sampling. This is good because it allows us to calculate the proportion, so on average, how often these specific behaviors are happening. The cons is we cannot calculate the true frequency or true duration of behaviors in this way, because if spawning happened, we'd give it a 1, but if spawning happened 7 times, we'd also give it a 1. So we're not counting the true frequency of behaviors. We're just counting if it did or did not happen in this 1-0 sampling. So we tend to overestimate duration, we tend to underestimate frequency, and it's difficult to determine the order in which these behaviors are happening because we're just recording 1s and 0s. To give you sort of a visual of these different types of point sampling, you can see here instantaneous point sampling will tell us, from when we start to when we end our sampling period. Let's say it's 10 minutes and we're going to look every minute. With point instantaneous sampling, we'd say, okay, at 1 minute, what behavior are they doing? At minute 2, what behavior are they doing? At minute 3, what behavior are they doing? At minute 4, what behavior are they doing? And we'd record it in that way for the whole 10 minutes, and we'd get 10 measures. With 1-0 sampling, we'd actually be watching within these intervals and then determining as the 1 minute mark hit, did the behavior happen within this interval? Did the behavior happen within this interval? Did the behavior happen within this interval? And so it gives us a sort of a different measure. You can use all combinations of these ways to sample and record your data. So we went through these different ways of sampling: ad lib, focal sampling, scanning, specific behavior sampling, watching for specific behaviors, and then you can record those in a continuous or in a time sampling way. These can all be used within your study. Don't think you have to just pick one. The study I'm currently working on, we do a lot of continuous recording on a focal individual. So I have a focal fish who's on a territory. We're watching him for 10 minutes, and we're continuously recording all the behavior that he is doing. But also, we are behavior sampling. We're watching the 15-minute video looking for a specific behavior and its occurrence. So we watch sort of the whole video, even when we're not necessarily looking at the focal fish, because we want to record when specific behaviors are happening. And we're actually also doing some instantaneous sampling where every 2.5 minutes we pause our recording because we want to get a measure of density. And so we're looking at density every 2.5 minutes. So every 2.5 minutes, the beep goes off and we say, okay, how many fish are in this given area at this given time? Even though that fluctuates throughout the entire continuous recording of behaviors that we're doing. So we're doing that's a scan sample that's happening instantaneously. I'm doing focal fish sampling, looking at one individual continuously. So you can see that it's a combination of all of these things. And your research question will lead you to which one would be best for you. So I know you're thinking, which one? What should I pick? That is really up to your hypothesis, your variables, and the information that you're trying to collect to determine if your hypothesis is going to be supported or rejected. So really it's up to you, all the different ways in which you can collect these data, which one would work best for you. So with all of that said, in our next class on Tuesday, I'd like to take the opportunity to discuss with everyone any questions that you have about possible ways to record your data, the types of data you might collect. Will you collect duration or frequency? Will you do focal sampling or scan sampling? Will you do continuous data recording, or will it be intervals? Let's talk about that. So bring your notes with you so we can talk about that, because don't forget, next Thursday, the 22nd, your draft research proposal is due. Within that draft, which is going to be a few pages long, you should be stating your hypothesis, giving me the null hypothesis and any alternative hypotheses that you have. Remember, a null hypothesis says there's no relationship between the variables. Alternative hypotheses can suggest a relationship, that one influences the other. One variable influences the other variables. And then you need to define your variables within the proposed research section. So clearly define what your independent and dependent variables are. If you are looking at behavior, you should be working on that ethogram. It doesn't have to be set in stone right now, but you should be working on which behaviors you're going to focus on and defining those behaviors. If you're not looking at behaviors, you don't have to. It's not a requirement. If you're not looking at behaviors, you do still need to define your variables and how you will measure those variables, whether they're behavior or not. For example, I will be looking at cortisol levels in my research proposal, which is not a behavior. I don't have to define that, but I do have to define what I mean by cortisol levels. I'll be looking at excreted cortisol levels measured in picograms per liter. Again, I got to figure that out. I'm working on it just like you are. So I'm going to figure that out, how I'm going to actually measure my variables. You need to start thinking about what information you'll have to provide to your readers, me, and your audience about how you'll actually do your study. Will you be doing continuous sampling? Will you be doing interval samplings? Will you be looking at, this behavior, that behavior, and how are you going to set that up? Is it going to be observational? Is it going to be experimental? Start thinking about all the information that you need. If you are unsure of what you want to do, that's okay. Don't think that I've said this already, don't think that you need to invent something novel that's never been done before. Start looking at previous work in your species or in closely related species or in sort of a design that you might be interested in, and see how other scientists did it. Borrow their ideas. That is encouraged in science. If someone else has already perfected an ethogram, if someone else has already perfected how to watch a specific animal in a specific way, you can borrow those ideas. Just make sure you cite them. Just make sure that you're providing the reference, the paper, where you borrowed those ideas for. And I'll be honest, you can never directly copy from someone else, not just because you shouldn't plagiarize, but because your specific hypothesis is going to be different. And so you're going to have to modify the behaviors, the methods of how you will collect that information. So it's not simply a cut and paste, but you can base your methodology off the methodologies of others. And this is encouraged. It is not frowned upon. Again, just make sure that you're citing that work. And of course, whatever work you cite, make sure you include that in your reference cited portion. So these are the sort of three areas of the draft research proposal that need to be written for next week. There is no article summary next week, so you don't have to worry about that this weekend. There is that knowledge celebration quiz in quotation marks, that is due by 9 p.m. on Thursday. Make sure you submit what your hypothesis is and your variables. That's right on D2L. Due by 9 p.m. Thursday the 15th. again, no live session. You're just watching this video. So no need to hop onto Zoom this week. We don't have any awesome researchers coming to talk to us. Just watching this video, finishing your knowledge celebration, no article summary, so that you can focus on your draft research proposal. With any questions, you can always email me. You can set up a private Zoom session if you'd like to ask me questions that you don't want to bring up during our normal class time. I totally understand. It's difficult with so many people in the room or on Zoom to ask those questions. So if you want to set up a time and meet with me one-on-one, I encourage you to do that. Just drop me an email. Or again, on Tuesday, we'll have time in class to discuss your ideas and your questions. I hope you have a great weekend, and I look forward to reading those draft research proposals. Have a good one.